Okay, so there are two main kinds of fermentation, alcohol and lactic acid. Now with alcohol you also get CO2, but with lactic acid you just get the lactate, no CO2. Now, every time it wiggles I worry.
You're making me nervous.
Here, we'll just like as my next.
I know.
Okay, so in alcohol fermentation, now pyruvate is going to go into fermentation. So in alcohol, it's 2 steps, and you get CO2 plus ethanol. Yeast can do this. So when you use yeast to make your bread rise, the reason it rises is because it's CO2, and there's alcohol also. So if you taste bread dough, you can taste the alcohol in it. So could you get drunk by eating bread dough?
No.
Actually, yeah.
Could you?
You'd have to.
You'd kill yourself.
What does that
on
mean?
bread.
You're gonna get sick before that happens.
You'd get a stomach ache because of the CO2,
Oh.
That seems like a
But
really
yeah, you do.
silly thing.
So the reason it's used in brewing and winemaking is because of the alcohol. So yeah, it does produce a significant amount of alcohol. Now, you do need to know that oxygen is not the final electron acceptor in fermentation. But I won't ask you what is. Just know that it's not alcohol, or not oxygen, rather. So they have a different final electron acceptors. And the other difference is you only get 2 ATP per glucose, where in respiration you get up to 32. So it's not nearly as efficient. But if you can do it, then at least you can get some ATP. So what cell in your body can do this? Well, it definitely is not the CO2 and alcohol, because you would blow up.
What
Okay.
cell doesn't receive oxygen in the body? Oh, are we talking about the cycle? I'm confused.
No, we're talking about what cell in your body can do fermentation.
Your
Your
muscles.
guts.
Your muscles are.
Okay,
The large intestine.
you're a caveman, and a saber-toothed tiger is chasing you, and you're going to be lunch for that saber-toothed tiger. And you're breathing in as much as you can, you're doing aerobic respiration as much as you possibly can, and he's gaining. So what cell can then switch into fermentation to give you a few more ATP, so maybe you can get away from the saber-toothed tiger? Muscle. Now, what happens the next day if you are not lunch?
You're sore.
You're sore because of the lactic acid.
Now, in the book, and it says that it's not necessarily the case.
The soreness?
About the whole lactic acid being the case in soreness.
I know that's kind of a
Actually
point of contention right now. Some say that it's because of muscle damage, tearing, that you're sore, but most biologists still think it's because of the lactic acid. And then that's broken down by the liver. But that's again, it's an argument that's going on right now. And so
So you're not going to test us on it.
I won't test you on why you're sore. But it's either because of lactic acid or it's because of the muscle damage. And the opinion is kind of going more towards muscle damage. So I don't know on that. I don't think anybody knows for sure yet. They're still working on that. Now, you have both obligate anaerobes and facultative. Obligate means they can only live without oxygen. Oxygen is poison to them. Facultative means it can go both ways. So your muscle cells, facultative. Yeast, facultative. Can do either aerobic or anaerobic. Now, an obligate, an example of obligate are Clostridia. Clostridia it's a genus, Clostridium, of bacteria that are gram-positive frauds, nasty little critters. what they do, if they are not in, if they're in the presence of oxygen, they will form a spore wall around them. And these walls are just impervious to drying out, they're impervious to heat. And so Clostridia lives in the gut of a lot of animals, especially wild animals. And so in the fecal material, you have these, the bacteria, which will then make a spore. Those spores can live in the soil for hundreds of years until they get in an environment that doesn't have oxygen, but it does have food, and it does have warmth and moisture and everything, and then they break down the spore wall, and then they have a party. So there are three main species in this genus of Clostridium. There's Clostridium perfringens, and I won't ask this, so you don't need to write down. It's kind of fun here.
I'm like, how do you spell?
And Clostridium perfringens causes gangrene. Clostridium tetani, what do you think that causes? Tetanus. And Clostridium botulinum? Botulism. And so if you have a deep puncture wound, you have to go in and get a tetanus shot because it's deep, no oxygen in there, food, water, nice and warm, let's have a party. And they produce an exotoxin, which means it's released. It's the exotoxin that causes tetanus. So that's why you get tetanus shots. Now with Clostridium botulinum, again, these spores are in the guts of wild animals, and so you grow veggies in soil outside, and those spores may have been there for a couple hundred years. And so you wash your carrots, but you don't get every single spore off from them. And so you put them, you're going to can them. So you put them in a jar, put them in a boiling water bath, and you cook them for however, time for the altitude and everything. And what happens during that is that you are forcing all of the oxygen out of that jar. And then when it cools down, what happens? Seals, right? So it's anaerobic in there. Now these spores have food, they have moisture, they don't have oxygen. Let's have a party and produce a lot of botulism toxin. Now botulism toxin is the most lethal natural toxin known to man. It is estimated that a teaspoon of it would kill everybody in the United States. So before you get Botox, you think about that. And what botulism does is it shuts down the central nervous system. So if you get Botox, you're getting a very low amount of it, not very much. However, there were 3 patients in Florida that did get Botox, and it shut down their breathing. So they're still on life support while their family's deciding whether or not to sue. So that is an obligate anaerobic. It can only live without oxygen. And so there's oxygen, makes them spore. So how do you get around that? I mean, you can buy canned carrots. So why are they safe? Well, they cook them at a higher temperature. Now you can do it at home as long as you use a pressure cooker because you're increasing the atmospheres inside of a pressure cooker. So at this atmosphere, the water, well, even here water boils at 80 degrees Celsius, but at sea level, water boils at 100. Well, that's not enough to kill the spores. But if you can raise it to 140 Celsius, that will kill the spores. And so if you increase the pressure inside, then the boiling point goes higher, and then you can get the higher temperature enough to kill the spores. And so when you buy commercially canned things, they have done it with a higher temperature, so it's killed the spores.
So you're telling me not to plant vegetables in my yard?
Just eat them raw. Because the spores aren't going to kill you. It's not until they have a party and produce all the toxin.
After I eat them?
They won't do that. You digest them fast enough. They have to have a while to incubate in there.
Okay.
See, that's why with a deep puncture wound, it puts the spores in there, and it, before you start getting symptoms of tetanus is about 2 weeks. And so it takes a while for them to come out of the spore stage, and then they have to multiply, and then they start producing the protein, the toxin. So it's not going to happen in your gut.
We'll survive another day.
You'll survive another day. Okay, so pyruvate is the common thing for both fermentation and aerobic respiration. So glycolysis is going to occur in both. Now, fermentation occurs in the cytosol, so anaerobic is in the cytosol. Now, you've got some intersections here so that you can go, it's catabolic to go to ATP production, to go that route. That's catabolic because you're tearing down glucose, and using that energy in the bonds to drive the ATP synthase. But you can also make anabolic things, so you can use the construction. So you can take these products and put them together. If you've got enough ATP, you're going to take the glucose that's left over and make glycogen and store it in the liver and in the muscles. Now, to go through this whole route, nobody I know of just eats glucose. So we are talking about, we've talked about glucose, but you eat pizza and you eat steak and cheese and all those things. And so you have to have a way to plug those things in someplace. Now, beta oxidation, I did not put a star in this, but just kind of remember beta oxidation. It's a word you're going to hate in physiology because it tells you how you break down fats to get them into here. And it's quite a process. It's quite a chemical process. It's about as bad as what we've learned already here in this chapter. Now, this chart, become very familiar with this. A few years ago on the department final, they said, where do fatty acids fit in? So that was rather a nasty question. I was really glad I had taught my students about it. Because some of the classes didn't. They didn't do too well on that department final. So fats are broken down into glycerol and fatty acids because remember, that's how you make a fat. You take a glycerol and 3 fatty acids. So we're going to chop it up. The glycerol is fed into here at glyceraldehyde 3-phosphate because if you went back and looked at that molecule, it looks like glycerol with just a phosphate stuck on it. And then we're going to take the fatty acids by beta oxidation, which again you will learn in physiology and learn to hate, which you're going to cut off these things 2 carbons at a time. And that's going to be fed into the acetyl-CoA conversion. So converting pyruvate to acetyl-CoA. We're going to feed those into there. Now, amino acids from proteins. First of all, we're going to chop the proteins up into amino acids. Now, those amino acids can then be reassembled into bigger proteins by your body. That's anabolic. Or we can use them for energy by, depending on what kind of amino acid, right here, or bring it down into the acetyl-CoA conversion, or bring it directly into the citric acid cycle. But you have to cut off the amino groups, and the kidneys excrete those. So this is one reason why Atkins diet's not a really good thing. It's hard on you because your kidneys have to handle all those amino groups. It has to make a lot of urea. And that's, takes quite a bit of energy. So it's much better to have a well-balanced diet.
Is that kidney stones?
Pardon?
Is that kidney stones?
No, kidney stones are uric acid, a little different process. But uric acid does have this on it, so, but I wonder, it could have something to do with it. Actually, it would. Thinking on to the physiology part of it, it would. It would have a definite. So if you have kidney problems, sometimes they will tell you to cut down the amount of protein that you eat, because again, it's kind of hard on about that. See if it would really make a difference. Okay, so this is all your other foods that you eat. Now, carbohydrates, if it's just starch, it's made up of glucose, and that's just, you chop it up and feed it into here. Sucrose is made up of glucose plus fructose, and so the glucose goes here, the fructose you put in here to the second step of glycolysis, which is fructose 6-phosphate, well, the third step. And so you bypass the first two steps, you just put a phosphate on it. So, yeah, this is all that's involved. Now, remember this for one of the questions I'm going to ask on the questions that I ask you afterwards. So, kind of remember this chart and realize that fats and proteins go in below, most of them go in below the glycolysis step. Okay, so we can use excess ones to make, again, bigger molecules. Now, feedback inhibition. By the way, going back to fat, you get twice as much energy from a gram of fat as you do from a gram of glucose, because fats usually have 14 to 18 carbons in that fatty acid tail. And so if you chop them off 2 by 2, and each one of them goes into acetyl-CoA, then you get a lot more ATP out of a single gram of fat than you do glucose. So when we have enough ATP, we don't need any more, we're going to do this thing called feedback inhibition. And ATP itself goes back here and inhibits phosphofructokinase. And AMP stimulates it. So what kind of an enzyme is phosphofructokinase? Allosteric. Exactly. And so this binds at a site away from the active site and halts it. And then when you run out of it, this thing, AMP, saying, hey, we really need some phosphates put on here, it's going to come here and bind at a site away from the active site and open all of them. So that is allosteric regulation. Okay, now. For the next midterm, this is the preschool version of cellular respiration that I expect you to know for the next midterm. Glucose is converted to what? Pyruvate. This happens in the cytosol. And what's the process called? Glycolysis. And NAD plus is converted to NADH, and that is reduction. And the electrons for this come from glucose. You also have ATP produced, and this is by substrate level phosphorylation because it's an enzyme reaction. Now, if you do not have oxygen, you can go the anaerobic route sometimes, and that's fermentation. It also occurs in the cytosol. And there are two kinds, ETOH ethanol plus CO2 or lactate. Okay? Now, with this, you have to have oxidation. Like with yeast. if you didn't convert this back to NAD+, you couldn't keep on going. And yeast will do this a whole bunch. Okay? Now, if you do have oxygen, then it's aerobic, and you can go into the mitochondria. Now, the mitochondria, inner membrane is convoluted. So this is the intermembrane space, intermembrane space. This is the matrix. Now, pyruvate is converted to acetyl-CoA in the matrix, and you have given off CO2, 2 of them. You have some NADH that's formed. And then the acetyl-CoA goes into the Krebs cycle, or citric acid cycle. And you have 4 CO2s given off. You also have 2 ATP, which is substrate level phosphorylation. And NADH, FADH2, which goes over to, let's change this bit, the inner, to the electron transport chain. That's proteins that are embedded in there, in the inner membrane. And that's the electron transport chain. Now, there, you're going to take the electrons off from these guys, and so you get NAD+, FAD, and that is oxidation. In the process, you get 30 to 32, no, 26 to 28. 26 to 28 ATP, and that is oxidative phosphorylation. You also get water. Okay? That's preschool version. So it's not in your book because what I did was I just put everything there that you need. Now the reason I didn't give you out this before is because you wouldn't have paid as much attention. Now on the next midterm, you may get a blank piece of paper, or you may get one that is partially filled out, but you will need to know every single thing that's on this.
Yes.
Now, it's color-coded, and keep it, because I always get emails from past students that say, oh, I lost it, I need it for physiology, please email it to me.
I was like, just gonna take a picture of my phone after. Sorry.
I asked you a question.
Yeah.
What exactly does phosphorylation
You need one more?
mean?
Phosphorylation, that means putting a phosphate on. Yeah. So with this, you're putting a phosphate on ADP to get ATP. So that's phosphorylation.
Actually makes sense.
Okay, so if you two will come over here, we'll do the questions. Now the deal is, if you get them all right, you don't have a quiz tonight.
Okay, come on, guys.
But since it's iffy, I do have it.
We got it, guys. It's cool.
Ready? Okay, good. Hi, Randy, you gotta come down here with them, you gotta talk to them about it. Did you have to work on it as a group?
Yeah, we're all.
Okay. For this, for these questions, you're going to have to use the diagram I gave you. Now, if you notice on that diagram, it's got numbers. Back in the little corner, you need your book, you need your brain, and you need your voice box because you have to talk as a group.
Oh, this is a question?
Okay, I want numbers.
So, two ATP.
I'm going to find out. Okay, so these are the things. So what you want to do is write down a column with these things on it so you can put the numbers by it. Okay. So this is the products.
That was somewhere. So mine.
Right, so are we going through and saying, in glycolysis, this is how much is produced in each one of those?
Yeah.
And then we'll go do the conversion of acetylcholine.
I know. I was like, oh, I gotta start on the side.
Okay.
Okay, so
So glycolysis.